So going back to this Michelson and Stars experiment, so semi-conjunctive replication mechanism. So how does that work? Again, so N15 is heavier than N14, right? So the original DNA strand should be labeled with N15. Okay, and then when cells are switched to N14 medium, so now when they duplicate, they will make a DNA strand that's labeled with N14, right? So when the first copy of the replication is made, making the first copies, so those copies will be the hybrid because the original DNA is N15 labeled, but newly made DNA is N14 labeled, right? So it depends on how they are mixed up in this daughter DNA strand. Okay, the newly made band, the pattern will be different. Okay, so that's the original DNA band, and the newly made copy band. Okay, where would they show up? Would they show up above there, or somewhere below at bottom, or above there? So that location of the band depends how the replication is made. Okay, so going back there, so what possibilities are there? So if it's the conjunctive replications, you will have the original DNA intact and then newly made DNA as a separate entity. Okay, and you will have keep going there like that. Okay, so if that's the case, then the original DNA should show up as the same location, but that didn't happen. Okay, you only saw one band, okay, at the kind of above lighter position. So what does that mean? The lighter position means at least it's this way. So if you have a semi-conjunctively copied DNA, they will be lighter because it's half N15 and half N14, and those two daughter strands will be identical. So they will end up on the same position, right? So that's what they saw. So those two DNA were lighter, so they located above, and then there was no difference between those two guys. They were in one spot. Okay, and is it not this? If it was this, possible. I mean, they will be lighter because they are mixed up dispersely. Okay, so they may show you the same location, but then how about on the next generation when the replication happens again? Okay, so if it happens again, if you give more time, what happens on these guys? Okay, so what they found was this band staying there, another band is showing up above case. Okay, so then if you look at that result, how do you explain that? Would it be this? Okay, so if you make copies of this semi-conjunctively, what do you see here? One DNA will be labeled with, the original, and the other DNA will be hybrid, original and hybrid, right? That's why you see two patterns out of these four. Two of them will be lighter, okay, and the other two will be kind of heavy because they are hybrid, right? So that's why you have two bands showing up there. Does it make sense? Okay, so if you do that again, you will see more of these guys, lighter bands showing up there. Okay, so it explains this. If it was this, okay, then you will, no matter how many times you do, that band will stay the same. Okay, you're not going to see that band appearing. Okay, so by looking at that result, it strongly says that yes, indeed it's this replication, not this or not that. Okay, so that was the finding. So DNA replication, when it happens, it is happening through semi-conjunctive way, meaning always one strand is coming from the original DNA and the other strand is a copy, newly made copy. Okay, and another experiment that was important. So let's summarize what they know about DNA. So DNA is the genetic material. They discovered that through several major experiments, right? And then so DNA can determine, okay, if it is used to make proteins, what amino acids will be used to build the protein molecules. Okay, so that's the rule. Okay, and then DNA cannot be the direct template for amino acid because they do not exist in the cytoplasm where protein synthesis happens. What does that mean? You know where DNA molecules are kept inside of cells, right? They are always located inside of nucleus. Then, how about the proteins? Where are they made? Normally it's the cytoplasm. So then, how is it possible? The DNA is moving out of the nucleus and be used as a template to make proteins? No, that doesn't happen, right? So you need to have some kind of intermediary molecule that bridging the gap between DNA and protein. So what is it? That's probably RNA molecule, right? So there are a bunch of RNA molecules you can see in the cytoplasm, and they have very similar looking as DNA. So now, after understanding DNA is the genetic material, then they understood what would DNA RNA molecules do? Would they be the bridges between DNA and protein? So they jumped on studying RNA molecules from that time on. Okay, so that's the importance of RNA molecules. So so-called central dogma. Okay, so Francis Crick came up with this idea. Okay, so DNA molecules are the important genetic material, of course, right? And proteins are, again, important molecules building up your cells and bodies. Okay, how do you bridge the gap? Okay, it must be RNA molecule in between because it has a similar structure as DNA. It can even pair up with DNA, okay, through a transcription process. And then must be RNA molecule is going out of the nucleus and to the cytoplasm, and then where it can serve as a template to build a protein. Okay, so that is the central dogma. Okay, so RNA molecules, what kind of RNA molecules are out there? It turns out there are a bunch of RNA molecules you can see inside of a cell. There are so-called tRNA molecules that are functioning in the translation process. About 10% of cellular RNA molecules are made up with tRNA molecules, okay, and amino acid molecules are attached at the tail of the tRNA actually, and then they have anticodon. They can bind on the codon of the mRNA molecules. And there are a whole bunch of other RNA molecules like ribosomal RNA and small nuclear RNA. There are so many of those. About 85% of the cellular RNA molecules are made up with these kind of RNA molecules. Okay, ribosomal RNA in different species, they do not reflect the difference in the DNA sequence, therefore not serving the template role. So these RNA molecules are not involved in the translation process. That's what it means. Okay, they must be serving something important, but something different, some different roles. Okay, so discovery of mRNA using bacteriophage T4. Okay, what does that mean? So what this experiment shows is, so as I talked about here in the central dogma, they were testing this idea. Are there really RNA molecules that are bridging the gap between DNA and protein? Okay, are there some RNA molecules really made as a copy of DNA, and then are those RNA molecules being used as a template for building protein molecules? They are testing that idea here using this experiment. So they use the bacteriophage T4, okay, which can infect E. coli cells. Okay, and then they traced it, what kind of molecules are being made from T4, unique DNA. So bacteriophage T4 is a DNA genome carrying a bacteriophage, so they can inject their DNA into E. coli cells. Once the injection happens, they realize that the normal E. coli cells replication doesn't happen, or transcription doesn't happen. So there's got to be the RNA that's made from the T4 DNA, and then which will be used to make the proteins that are needed to package the newly made T4 bacteriophages. And then at the end, when T4 bacteriophages are released from the cell, they are completed bacteriophages. So they try to isolate the RNA molecules that are made from the T4 DNA, which will bridge between the T4 DNA and the proteins that are made from T4. Okay, and they were able to isolate those T4 RNA molecules from the host cells that are being infected by T4. So those RNA molecules, indeed, when they isolated, have the matching sequence as the T4 DNA. So they found that bridging molecule, the mRNA molecule, through this experiment, the T4 bacteriophage experiment. Okay, so indeed they they illustrate that, yes, indeed the RNA molecules are bridging the gap between the DNA and protein. So the transcription into mRNA molecules, that's what they found. Okay. And then one more important experiment that was done by Nirenberg and Mathai in I think it was 70s or 60s, but I'm not sure. But that's the experiment that elucidated the triple nucleotide codons that code for specific amino acid during the translation. Okay, so what they did was they used the multiple new nucleotide which is making up mRNA sequence, and then they did in vitro translation using the cell lysate, bacterial cell lysates, and then radioactively labeled amino acids were added, and they were able to identify, if we have multiple use, which amino acid will be made in the in vitro translation system. They identify that if you have multiple use, you can make bunch of phenylalanines. Okay, so by combining different nucleotide sequence making up mRNA, they were able to identify different codons that will designate different amino acids. So that is a novel experiment that identified the codon tables that was done by Nirenberg and Mathai's experiment. So at the end, so we study all these things because of the studies done in the 20th century. So all those findings showing that yes, DNA is indeed the genetic material, and then RNA is made off of DNA, and then RNA will be used for the transcription as a template molecule. So that's the breakthrough of the modern molecular biology. And then genomics, the study of genomics. These days, genomics are important because that's the study that determines the complete genome sequences of the variety of organisms. So more and more genomes being mapped out and sequenced out. So we use supercomputers to store the genomic sequence of DNA molecules, gene sequences, and all those things. Okay. So there are two terms describing genomics versus genetics. They are different, right? Genetics is the study of, I guess, heredity, like Mendel's genetics. Okay, genes are referring to the specific sequence of DNA on a single chromosome, and gene is a functional unit that gives you some kind of phenotype and the heritable unit. But when you say genomics, it's the study of the entire organism's genes, entire chromosomes. Okay, genome refers to organism's entire genetic makeup, and those genome is made up of thousands and thousands of genes, and you also study the gene sequences and everything. All right, so that's the end of lecture 1. All right.